Namaskar, welcome to PK's classes. Today we are going to study about the cholinergic receptors. So when we say cholinergic, the neurotransmitter is acetylcholine. So when acetylcholine is the neurotransmitter, in short we say it ACH. It acts on the receptors which are named as cholinergic receptors. So the receptors on which acetylcholine acts as the neurotransmitter are called as the cholinergic receptors, and they are of two types: muscarinic, because the alkaloid muscarine acts as agonist on those receptors, and nicotinic, because the alkaloid nicotine acts on those receptors. So muscarinic receptor is again of 5 types: M1, M2, M3, M4 and M5. Nicotinic receptors are of 2 types: NM, NN. NM, M stands for muscle, that is the skeletal muscle, and N stands for neuron. So neuronal, neuron in ganglion, the mass of cell bodies outside CNS, and in also CNS it is located. So the nicotinic receptors, we also call them as nicotinic acetylcholine receptor, and these receptors are ligand-gated ion channels. Ligand-gated ion channels or ion channel linked receptors. These muscarinic receptors M1, M3 and M5, these 3 receptors are GQ type. GQ type means the effector is phospholipase C, and the second messenger from PIP2 by the action of phospholipase C, there will be formation of IP3, inositol triphosphate, which increases the level of calcium. So the second messenger is inositol triphosphate. The other 2 receptors M2 and M4 are of GI type. So here the effector is adenylyl cyclase. So since it is GI type, it inhibits the enzyme adenylyl cyclase. So it decreases the level of cyclic AMP. So the muscarinic receptors act through the second messengers and G protein coupled receptor there, whereas nicotinic receptor, these 2 nicotinic receptors are ion channel linked receptors, and mainly they are linked to the cation channels, that is the sodium, potassium and calcium. So you can note it down if you want the notes. Cholinergic receptors are proteins that respond to the neurotransmitter acetylcholine, which is involved in various physiological functions such as muscle contraction, heart rate regulation and cognitive processes. So cognitive processes mostly in CNS, and also the heart rate and muscle. Mainly muscle, heart and glands. These are the 3 areas where the muscarinic receptors are located. So 2 types of cholinergic receptors, muscarinic and nicotinic. Muscarinic again 5 types and nicotinic again 2 types. So muscarinic location mainly found in heart, the smooth muscles and glands, and they mostly mediate the effects of acetylcholine in the parasympathetic nervous system, and they play a key role in heart rate, digestion, and other involuntary functions, mainly involving heart, smooth muscle, and glands. Then the subtypes, 5 types, and already discussed, they are all G protein coupled receptors, and M1 receptor linked to GQ, which activates the phospholipase C and ultimately increases the calcium levels through IP3, inositol triphosphate. Then M2 receptors, they are primarily found in heart. So M1 in addition to the central nervous system and autonomic ganglia, M1 is also located in GIT, where it regulates the acid secretion, gastric acid secretion. So M1 antagonists like pyrangyepin, telangiectin we can use in ulcer, treatment of ulcer. Then M2 primarily found in heart, where they play a role in reducing heart rate, producing bradycardia. They produce bradycardia and decrease in heart rate because they are coupled with GI proteins which inhibit the enzyme adenylate cyclase and decrease cyclic AMP. For details, you may refer to our earlier classes on M2 receptor, M1 receptor, M3 receptor, nicotinic acetylcholine receptors, and the links are provided here. Then M3 muscarinic 3 receptor, and it is again GQ type, and mostly they are involved in smooth muscle contraction. So smooth muscles of gastrointestinal tract. So GI motility is regulated by M3 receptor, and they also regulate the secretions from the glands, glandular secretions. Then M4 mainly found in CNS and mostly involved in movement and cognition, and coupled with GI proteins leading to inhibition of adenylate cyclase. M5 receptor is coupled with GQ proteins like M1 and M3 and located mostly in CNS, and they also modulate the dopaminergic system, M5 receptors. So GQ protein is coupled, activates phospholipase C, forms IP3, and IP3 mediated calcium release occurs, increasing the intracellular calcium. Nicotinic receptors found at the neuromuscular junctions in skeletal muscles and also in the autonomic ganglia, both sympathetic and parasympathetic, and in CNS. These are the 3 areas. Then the nicotinic receptors mediate fast synaptic transmission. They are involved in the initiation of muscle contraction because they are located in the neuromuscular junction, and in the ganglion they help in transmission of nerve impulses. So these are all ligand gated ion channels. So when acetylcholine binds, there will be opening of the ion channels, and there will be full flow of cations such as sodium, calcium, and potassium. Suppose this is the nicotinic acetylcholine receptor, and the cation channels are linked. So mostly the cation involved with sodium, and there can also be influx of calcium, and there can also be outflux of potassium because potassium is more intracellularly, sodium and calcium are more extracellularly. But the net action is that of sodium. So out of these three, the major action is that of sodium. So net action is sodium mediated depolarization. Sodium causes the depolarization. So the net action is depolarization of the cell. And nicotinic acetylcholine receptors based on the location can be muscle type and a neuron type. So NM, nicotinic muscular. And again, it has 5 subunits. All the nicotinic acetylcholine receptors have 5 subunits, and again they may be homomeric or heteromeric. And muscle type are mostly homomeric, where all the 5 subunits are alpha 1 mostly, and the nicotinic neuronal type is heteromeric, containing both alpha and beta subunits. And in addition to them, there can also be other like delta, epsilon, other subunits can also be there. But mostly muscle type is alpha 1 containing, found at the neuromuscular junction, and the most common one contains the 5 subunits, where 2 alpha 1 subunits, 1 beta, 1 delta and 1 epsilon subunit are there. And for more details, you can refer to the nicotinic acetylcholine receptors. And when the acetylcholine binds to the receptor, the channel opens, allowing sodium to enter into the muscle, leading to depolarization and hence muscle contraction. The nicotinic neuronal type or NN type of receptor, it is found in the ganglion, autonomic ganglion in the peripheral nervous system, and in the brain or CNS, central nervous system. And here mostly they have alpha subunits and beta subunits. So the 5 subunits are a mixture of alpha and beta subunits, and these receptors are involved in synaptic transmission, modulating the neurotransmitter release. So as I said, the nicotinic acetylcholine receptors can be homomeric or heteromeric, and if it is homomeric, say all the subunits, all the 5 subunits are only alpha, or heteromeric, 2 alpha, 1 beta, 1 delta, 1 epsilon in case of nicotinic muscular receptor, or a mixture of 2 to 3 alpha or 2 to 3 beta subunits, then it is called heteromeric. So thank you. For details, you can refer to individual receptors, or the link is attached here with. Thank you.